To welcome on stage Aaron Smith, founder of Unlearn.AI, who's going to give us some practical examples of how to leverage digital twins in clinical trials. There you go. Here is the clicker.
Great. Well, thank you very much for coming out and listening. I should mention I'm a co-founder of the company. There are a couple others, so I can't take credit for it entirely. So I'm going to talk a little bit about AI-powered digital twins, and the main point here is to present the key concept, how it can be applied, and then try to get to brass tacks and talk about a case study that is practical. So first of all, I'm going to introduce the concept of digital twins for disease modeling. I think it's important that I make it clear what I'm talking about when I talk about digital twins, because this is a term that's being used a lot right now. So it's absolutely essential to start out that way. Talk about a particular application of digital twin models to increasing the efficiency of clinical trials, and then talk about a case study involving adding efficiency to a phase 2 Alzheimer's disease trial. So a digital twin of an individual is a rich model of that person's clinical status. Digital twins ultimately provide answers to counterfactual questions about an individual. How will their disease progress? What will likely be effects of interventions? What are risks of significant health effects? How will they progress, and so forth? If you want to see this pictorially, it's very practical to see this picture here of a subject who is a person who has Huntington's disease. A bunch of measurements are made for that individual at baseline time. All these granular measurements related to their health, particularly related to Huntington's disease. And then the digital twin model can provide projections into the future, probabilistically, of how these are all likely to progress simultaneously. And the plus or minuses there indicate that these are actual distributions, and those are standard deviations. At Unlearn, core thing we do is build AI models that generate digital twins of any patient, powering important development applications. And the thing and these models are built on large data sources about patients' health, mostly clinical trials and registry data in particular indications. And we build a model we call a digital twin generator that allows you to, for any patient in that indication, create a digital twin for that patient and use it in various applications. So I'm going to talk about one of the most exciting applications of digital twins, which is increasing the efficiency of clinical trials. Digital twins can enhance the efficiency of both randomized and uncontrolled trials. I'm going to talk entirely today about the randomized control trial application. I want to point out that there's another application, which is probably something very natural that a lot of people have in mind, which is if you have a digital twin of a subject in a single-arm trial, you could simulate their potential outcome under control and thus have a simulated control or a synthetic control of that trial. That would be a single-arm application. That's not what I'm going to talk about today. I'm going to talk about an application to randomized control trials. So I'll start off by talking about the basic relationship between power and sample size. Many of you know this. When you're designing a clinical trial, you have something in mind. You have the minimum clinically meaningful effect that you like to detect. And you basically have two parameters that you can play with. You have two connected parameters that you can play with, which is the sample size and the power. You want to set a certain power level for your trial, which gives you the chance to reliably detect that effect. And that ultimately is going to be set by some parameters that you can measure about the data, like the variability of the outcome, and then you can set the number of participants to achieve the power that you want. That's the most basic description of that trade-off. And so you have a curve like this. The key thing here is that decreasing the variance of the treatment effect estimate in your trial is one way to add efficiency to your trial. It's one way to add power. There are a few other ways you could add power to a trial. One thing you could do is enroll populations with smaller variability, or basically you could enroll a subset of your population that has a better signal-to-noise ratio in your trial. Another thing you could do is use endpoints that have less variability, so that's another way that people optimize trials. But the thing I'm going to talk about here is you can increase the power or increase the statistical efficiency of a trial by decreasing the variance of the treatment effect estimate. And you can see here what happens when you have a change, when you can, say, move from the original design trade-off here between relationship between number of participants and power. If you can decrease the variance of your treatment effect estimate, you can move that power curve up, allowing you to achieve more power with fewer subjects, or achieve the same power with fewer subjects, or more power with the same amount that you have. So Unlearn pioneered an approach to incorporate digital twins into randomized control trials called PROCOVA, which stands for Prognostic Covariate Adjustment. This is something that we developed some years ago now. We've been through EMA qualification. FDA concurred with EMA's qualification in this case. And we've been through successful Type C meetings for integrating this approach into protocols of phase 2 and phase 3 trials. What it fundamentally involves is a way to enhance randomized control trials with predictions made from a digital twin model for each subject who's been enrolled in that trial. And ultimately, there are two major benefits of this approach. As I alluded to earlier, you can essentially add power to your trial and keep the original design, in which case you end up increasing the probability of success of your trial. On the other hand, you could prospectively reduce the sample size of your trial, maintaining the same power level, but decreasing the number of subjects. Typically, you'd reduce subjects from the control arm, and ultimately, you don't have to pay as much to run that trial. It's fewer subjects, and even more critically, you can get to market faster. And operationally, this approach is very simple. You have our digital twin model, which exists prior to the trial. It's pre-specified. At the start of the trial, everyone's baseline data is collected, and you can create the digital twins and convert them into prognostic covariates in this trial. And then the randomized control trial is run as usual. And then there's a statistical analysis at the end, which incorporates all the data that's measured in the trial, as well as the predictions from the digital twin model. So that's all the theory of the approach, and I want to talk about a particular case study and talk about the benefits of this case study. So here is an example of adding power to an Alzheimer's disease trial. This is one example of us applying our Alzheimer's disease digital twin model with a partner, AbbVie. This was last year. This was a case study we did with AbbVie. So they have a fully proprietary trial data set that we got to apply this approach on to prove its efficacy. So it's a phase 2 randomized control trial with about 450 participants in an early AD cohort, relatively contemporary, 2017 to 2021 is when this trial was run. I'll jump right to the results in terms of variance reduction, as I mentioned earlier. This is an analysis that affects all of the outcomes in the trial, by the way. It's not just your primary outcome, so you can increase power of all the secondaries as well. In this case, 4 different outcomes we're looking at, and there's 3 different dosing arms in this trial. The pink or the purple one there, if that is purple, corresponds to all groups. And what we're seeing here is the variance reduction of the treatment effect estimate between the most standard analysis you could do, which is no adjustment, versus adjusting for baseline, and then versus the digital twin-based approach. And you can see robust variance reductions across all outcomes simultaneously in this trial. So what does that mean? As I said earlier, you could use the gain that you get in this case in 2 distinct ways. You could just take power gain, and you could run the trial as you would normally, but just have a more powerful analysis, or you could prospectively reduce sample size. The benefit of adding power, or one way to think about adding power, is that you're increasing the effective sample size of your trial. It's as if you ran the trial with more subjects, but but you didn't have to. On the other hand, if you prospectively reduce the sample size, that's equivalent to moving left on this chart, where you reduce the number of subjects, maintaining the same power level. And so I want to talk about the numbers, what this looks like for a, this is actually based off a characteristic large Alzheimer's disease trial. These numbers come from actual validation metrics of our AD model. If you're planning a phase 3 AD trial, 1,600 subjects, 800 per arm. If your default design is to power this trial at 90%, you have a certain expected enrollment time, number of subjects being 1,600. If you simply were to add power with this approach, you would gain about 380 effective subjects for about a 5% absolute gain in power. On the other hand, if you wanted to prospectively reduce the sample size, you could reduce the sample size 280 subjects from the control arm, maintaining 90% power, and based on a model of recruitment time, you could reduce recruitment time almost 4 months in this case, which is quite significant. So those of you who know a lot about pharma or drug development know that these can correspond to some eye-watering numbers, which I'm of course going to show you right now. So if you consider, for example, in these first 2 cases, how much money you save by reducing the number of subjects in your trial simply in terms of what it costs to execute the development, you're looking at, in either of these cases, somewhere between $28 and $38 million. On the other hand, if you have a blockbuster in Alzheimer's disease, conservatively, you're looking at $50 million per month. So recruitment time is actually incredibly important. And for a phase 3 drug, for example, that is generally expected to be successful, although neuroscience is maybe a tricky area for me to say that in, you want to have as much time on market as possible, and that's really where an enormous amount of gain, of value gain comes in. So with that, I will leave it at the benefits page reprise. Again, the 2 basic approaches here, adding power or prospectively reducing sample size, have these important benefits that can be applied in phase 2 and phase 3 trials across the board. Thank you very much.
Thank you, Aaron. We have time for a couple questions.
That was great, thanks. Does this work equally well across any therapeutic area? Is it kind of agnostic to that?
So yeah, the methodology, generally speaking, applies everywhere, like the statistical methodology, but you have to build a model of disease progression, and different indications have different features, there's different availabilities of data, there's different types of endpoints, there's different disease state structures of diseases and so forth. So your mileage may vary, but yes, the general approach is applicable.
Could have time for one more question. Suhas.
One over there.
Alex, yeah.
Thanks. Hello. How long does it take to build a digital twin, and how do you validate that?
Yeah, it's a good question. We are getting better at doing it faster and faster. That's one of our key technological issues as a company, is we want to be able to do this really rapidly. So there's certain amount of effort. There's a lot of effort involved in building a harmonized data set of past clinical data that is useful for training a very accurate machine learning model. So there's a significant amount of work there. The actual training and evaluation is something that we do internally in a period of a month, let's say, or less in some indications. Validation is a more interesting question. So what we typically do is sort of industry standard data splitting, all that kind of nested cross-validation kind of thing for evaluating our models internally. But then we have held out clinical trial data sets that are just fully held out from the process that we use to evaluate performance. And then subsequently, what I showed here is a perfect example of what we typically do with sponsors, which is they will have an entirely proprietary clinical trial data set that they own themselves. And they just tell us to reanalyze an old trial and show us that it works. So that's the most powerful way to do it. And for example, in AD, we have many of these examples where we've done this with partners. So
Thank you.